maybe we can start while we're waiting. So I'm one of the PIs of the Kidney Precision Medicine Project, which is a NIH-funded effort to build a human kidney atlas in health and disease. And for this effort, we're obtaining largely kidney biopsy tissues from people who have both what's called chronic kidney disease and acute kidney injury, but also healthy reference tissue, which is coming from a variety of different sources, including living kidney donor biopsies and other sort of operative procedures. So different kinds of tissues are coming forward. And also we've been using a lot of tumor nephrectomy tissue where outside of the tumor, the rest of the kidney is relatively normal for working with a variety of different tissue interrogation methods in the sort of early phases of this study. So we've been spending a lot of time thinking about what's the purpose of the different kinds of tissues that we're collecting, how do they differ, whether it has to do with ischemia or freshness or the amount of tissue, what kinds of QC methodologies can we deploy, how can we harmonize metadata that goes around the tissue that's being collected. We spent a lot of time on pilot processing protocols as well, with a number of different tissue interrogation technologies ranging from single cell techniques to spatial resolution for metabolomics and proteomics within the kidney, et cetera. So those are our sort of broad-based interests in trying to define what's the best way to obtain tissue for our purposes and what's the best way to process and share these kinds of tissues. So maybe we have a small group here. Maybe we can sort of go around the table, so to speak, and ask everybody what their what consortia they're representing and what their interests are, and how these questions about tissue collection and processing are relevant.
I'm happy to kind of go next. My name's Lori Coburn. I'm at Vanderbilt in Nashville, Tennessee, and we're part of the Helmsley Gut Cell Atlas project. Our focus is on Crohn's disease and making a gut cell atlas, especially relevant atlas comparing Crohn's in the terminal ileum and ascending colon to healthy controls. And the tissue we're collecting includes both surgical resection specimens as well as colonoscopic endoscopic biopsies, which we are then parsing out, kind of similar to what you're doing for the kidney map, is taking fresh samples that are then going for single cell RNA processing and single cell RNA sequencing. We're getting whole, especially from the surgical resection specimens, getting blocks that we can do multiplex immunofluorescence on to then have more spatially relevant data related to the targets that we are finding at the protein level that we're finding from the single cell RNA sequencing. And then trying to compare those across different spectrum of disease, treatment, exposures, things like that. So looking at how we can keep up with how quickly we're able to get the samples to the different processing areas, and how that is affecting our data quality is one of the focuses that we've been looking at. What different media, et cetera, are we putting the sample directly into to give us more processing time on the back end from when we are in the operating room versus when we're in the endoscopy lab. So that's kind of a brief overview.
Right, I'm Jian Choi from NCI, and I'm kind of a newbie to this single cell field, but I'm mainly in lung cancer genetics field. So from that perspective, we are starting this single cell EQTL project in lung tissues, human lungs from 120 Asian people. So I wanted to be in touch with the bigger consortia, not only just lung, but if there's any interest in building the QTL data set out of this, because that'll be a great interest for us. We mainly do GWAS follow-up studies, and especially in minority populations like Asians. So I think we just started to collect samples, actually the planning stage. So it'll be a great learning experience for us, for our group. And nice to meet you all.
Thanks. And I see we have Stavros on the call. Stavros, we were just mentioning, we thought that the beginning of this session, rather than break out into the subtopics, we would have sort of everyone with the 3 different co-moderators in the same room. Is that Zoom room? Is that feasible or not feasible?
The other 2 groups, I've already visited with them, and they've already started working on their subtopics. Okay. So
we'll
probably,
keep going.
yeah. Okay. I'm going to put my email address in the chat. In case you guys need anything, I'll be bouncing back and forth between the 3 rooms about every 10 minutes. So that way, if you need anything, you have it.
Okay, thanks. So let's keep going and hear from everybody. What are your interests? And then we can get to what do you think are the key questions that we should try and address with very diverse needs amongst the different research projects and consortia that are in this chat room.
I can be the next. Hi, everyone. My name is Yuhua Zhen. I'm from Johnson Diabetes Center, Harvard Medical School. I'm part of the NIH-HCA consortium, and specific program my lab and my teams are focused on is adipose tissue, which is kind of like underrepresented in HCA. And we are still at the early stage. We are collecting tissues, but as everybody know, unfortunately, we have to stop the processes because the current situations. But I really hope to be able to join this meeting to hear from everyone's experience, because for X tissue is multiple places. And to us, one of the big challenge is the size of the tissue, and also the nature of the cells is really fragile. So we are currently proposing to perform single nucleus RNA sequencing instead of single cell, because the 10x cannot single cell the adipocytes. And so it's also then the tissue handlings and is tricky. So anyone has in experience or in your tissue that you encounter adipose tissue, I'd like to them from your experience. Thank you.
Hello, I'm Nasreen Haq. I am at the New York Medical College in New York, and what we are interested in working is in atherosclerosis. So what we have is we started a repository in which we collect the samples from carotid endarterectomy patients during the surgery. We're trying to map that as a progress for atherosclerosis. And our question, my main question is then how do we obtain the normal samples, for which we are looking at autopsies. And really this meeting would help us sort of form a plan how we would be doing these processes.
Thank you. So one question I have is, are you all collecting, you're all collecting diverse tissues under diverse clinical circumstances, some elective, some non-elective, some, you know, if you're missing, if you're not there in the operating room, you're not going to get your sample. It's quite variable. Are these all, are all of your plans single center tissue collections, or are you trying to collect samples from more than one location? And if so, how does that affect your your SOPs and your training of personnel and who actually collects the tissue and who processes the tissue. Maybe I'll stop here and see if you think this is an important area for discussion.
Yes, so our samples we collect are from our neighbor, that is Westchester Medical College, Medical Hospital. That is one center, but we also have in New York upstate another center. So the way that we work out is because all these hospitals are connected. So we are informed and the sample is sent directly to my lab when I need it. But after the first few experiments, I realized that it would be feasible to expand our studies because to see that what we are finding in our initial studies in the local hospital, is it also true for others? Because what we are looking for on one aspect is we're looking at immunology, but on the other, we are also looking at the microbiome. So what we have identified certain bacteria in these samples, we want to expand this and see if we see in other samples. So I think the biggest difficulty is organizing that kind of huge collaborations.
Right, and do you have a sense of how sensitive your tissue collection is in terms of timing and who collects it and time to initial processing and those kinds of questions? And
yes, we'll always face those. However, we have worked around it for the bacterial collection. It is not so difficult, but of course, the host cells, it becomes a little problematic, especially we want to do RNA-seq and other studies. But we have been able so far to get the gene expression that we require. And we are still working on it to sort of standardize the technology.
thanks. How about others?
I'll say for us, for our particular questions that we're answering, we're collecting only at the single site, either with our endoscopy lab, or we are fortunate to have a site of the Cooperative Human Tissue Network through the NCI at Vanderbilt. So they're helping us get the surgical resection specimens that allow us to be a little more involved from that standpoint. But we're also dealing with, as part of the Gut Cell Atlas set of groups with the Helmsley Charitable Trust, there's 11 projects in there, and the goal is to potentially share samples across sites, but definitely sharing raw data. So we're beginning that process of trying to, similar to what we're doing here on a much larger scale, figuring out how to format the data so that raw data can be shared for RNA sequencing or things like that, where it wouldn't necessarily be, or maybe wouldn't be as easily feasible to send a fresh sample or an encapsulated sample to another site to run the actual analysis, but to then be able to pull the data together. So it's a little bit of both, but specifically for just the actual tissue samples that we're processing, we're just using our single site at this point.
Yeah, thanks, that's helpful. Others?
So for us, it's single center based for now, but the major issue for us is that to get a bunch of Asian samples, we're getting these samples from South Korea, and then they have to be shipped to the US, and then that's a major challenge. And our collaborators are mainly physicians, and in the lab, in my lab, we're trying to optimize a protocol and then provide a SOP for them, and that's not going to be simple. And also, we're going to do the RNA sequencing for eQTL, and the standard protocol requires fresh tissue, but we're trying to do multiplexing because for eQTL, number of individuals is a power. So we are trying to multiplex 8 individuals in one 10x run, and that way we asked our collaborators to dissociate the fresh tissue and then freeze and then ship to us. So coordinating all that will be a
Right.
considerable challenge.
Yeah, and we have, in the Kidney Precision Medicine Project, we have sites all around the country, and these are biopsy samples, and it's really a small amount of tissue, and we would like to be able to do both digital standard histopathology, but also a variety of different, as mentioned, sort of spatial proteomics, spatial metabolite profiling, as well as single cell technology. So it's a real challenge because we find that if there's any variation in the standardization of the immediate tissue processing, it has enormous effect on downstream analyses. And so we had to go to great lengths to train pathologists and research coordinators to be there on site, and we made site visits and videotaped everybody, and every month they make another video of the processes so we can kind of constantly QC. So as simple as it seems, and you know, the people on this call, everybody has diverse tissue in diverse locations where tissue collection is going to take place. And you know, we all want to harmonize our data with other sets of data to sort of maximize the power and the utility of what we're doing. So at least for us, it's a big ongoing challenge, was a big challenge to get it set up and moving, but then maintaining the quality and consistency over time is also a big challenge.
Videotaping sounds like a very good idea. I think I should adopt that idea. And I have a question. Does anyone, has anyone tried optimizing protocol in, for example, mouse tissues and then translate that into human tissue? Because of course we cannot spare too many human tissue and for the purpose of optimizing protocols.
Might I nudge you guys? It sounds like you're getting into the meat of identifying some needs. So might I nudge you guys to go over to the Google Doc, perhaps select a scribe or two and start transcribing into that first yellow or green box that says part 1, where it's the how might we's. Does anybody need that link to the Google Doc again?
That would be great if you want to provide the link.
Yep, just popped it into the chat.
Okay, perfect. So if we can get a couple volunteers as scribes as we go along to just add the key elements to the discussion, that would be great. So can you go back and repeat that last question, because it was a good one?
Would anybody like to volunteer, just so we know who you are? We'd like to get at least 2.
We've got one volunteer.
Yeah, I'll be happy to volunteer.
All right. I won't interrupt your conversation any further, Jonathan. I'm going to hop out and check on a different group. Thank you so much.
Thank you. Okay, where were we? Because I remember it was a really good point or question that was being raised.
Yeah, I had a question about, like, in terms of optimizing protocol for human samples. Instead of wasting precious human samples, I was wondering if it can use mouse tissues or other alternatives, and then how translatable that is to human tissues. Because if it's a whole new optimization, then it's just wasted time. But I wonder if anybody had any experience with that.
You could spend a lot of time optimizing in mouse, but doesn't really come up to the same condition. So it's going to be very difficult. I mean, obviously you have whatever studies we are doing in the human has previously been done in the mice, and we have some standardization, but it doesn't definitely, it's not the same. You could, in an animal setting, you could design your experiment the way you want, but when you're getting the sample, who's sending you the sample, these are really factors that really you have to come for specific for human tissue. That's my thinking.
Yeah, we also have similar experience. We optimize, the condition. We perform quite a few single cell sequencing experiments using mouse tissues. It's a adipose tissue, and we using the mouse tissue. There's a lot of advantages. We could validate the data using lineage tracing experiment, back sortings, and so it's definitely we have more control. But to answer your question, I also feel it's not possible to entirely use the same tissue, the same protocol we apply in mouse tissue to the human. I think for the human tissue, because even it's the same type of tissue, the lipid composition in our case, especially dealing with fat tissues, it's still different. We feel that we still need to perform some sort of optimization to handle the human tissue.
Yeah, I think that's our experience in the in our project. You know, each of the tissue interrogation sites had to get through the peer review process had done a lot of animal model work and whatever their technologies are. And then we circulated nephrectomy tissue as pilot. We did pilot studies first with nephrectomy tissue from people who were undergoing tumor nephrectomies with the normal parts of the kidney, where we could get consistent tissue to give to each site to work with. But even then, you find that when you're working with clinical biopsies or surgical specimens, totally different. There's much more heterogeneity, variability, what you get from one biopsy to the next or one surgical excision of tissue to the next. And so what was totally optimized for mouse, let's say, and then totally optimized for nephrectomy specimens may not be as optimized for when you actually get, let's say, biopsies or surgical specimens. So, it is a challenge. You can prepare, but you will also sort of learn as you go along from your first specimens that you get, how to optimize whatever it is you're trying to do with the tissue. And the challenge, of course, with human tissue is we don't want to waste any of it. If we have altruistic patients that are participants in our studies, and they may or may not be undergoing some risk, but for this study, we want to maximize every single piece of tissue that we get for the greater good. So, how much prep work do you do, and when do you just plunge in? And then reassess every certain number of procedures, how to make it better. Those are, questions that I think we all wrestle with.
One of the things that we are trying to do is the surgeon or the pathologist will always save the specimen, a part of the specimen, and they use it for FFP blocks. They would make the paraffin blocks. And what we have been able to do is extract RNA from that. And so that would be a little more uniform because that is a systemic process they've been going through. So that is one way if you are just looking for that. But obviously where live cell is needed, then it becomes a problem. But we can correlate our data from both the paraffin and what we get from live cell samples.
So from FFPE blocks, extracting RNA from that, that'll limit your applications, right?
I know, yeah, but you at least have some, like uniformity. Because here you don't know when you're getting the sample, how long would they call you, then what stage you are in, But there they have a routine setup. So we're trying to get those samples as well. So that is an added, data that you can then correlate with this data. And the other thing is that what we have found is getting the FFP samples is easier because you can get it from the archive, whereas here live sample becomes you'd never know when you're going to get it. Limitations.
Nila Herdiner from the Anatomy in Vienna and Division of Anatomy, Medical University of Vienna. And we are using our body donor program to standardize some of the pathways for sample taking and imaging at the same time. I don't know if you can hear me.
Yeah, we can hear you, but speak up a little more if you can.
Okay, sorry, maybe that's better.
Yes.
I was saying that we are trying to use the body donor program we have in-house to optimize some of our sample taking procedures and including imaging in the pathways so that we can optimize that before we get the freshest possible body donor samples that we can then use to run through the pathway that we did.
Are you having difficulties with getting the body donors? Because we were thinking of getting the autopsy samples, but it all depends on the hospital and
Yeah.
what is the state of mind of, you know.
So for us, it's no problem to get the samples, but it is difficult to get them fresh enough to get good RNA data.
Yeah.
So 6 hours after death is the freshest that we get at the moment, but we're working on optimizing that as well in working together with the hospitals and the interment service.
Thanks. So are we capturing all this in our Google Docs? These are, I think important questions around how much optimizing to do before starting with human clinical samples and how can you do that? And this last, Anna, thank you, this question about time and timing and degradation of the sample, how much can you tolerate for which types of assays? Clearly transcriptomic data is going to be, well, I guess all kinds of data is going to be very sensitive to freshness of the tissue, and how can you optimize that, or at least understand what the parameters are.
Along with that, we had definitely talked about trying to get donor specimens to do some optimization. One of the things we worried about obviously was the timing of that and how that would affect our assays, but another thing we ran into is using donor services. At least that was the pathway that had been previously used by other labs. The cost for obtaining those specimens was quite a lot. It could be up to $500 for one section of the bowel. And if you wanted multiple sections, which we were interested in, you could potentially be paying up to $2,000 just to get the piece of tissue when you didn't know what the timing necessarily would be. So that also played a role in ours not using that as part of our optimization, because we didn't have that startup cost, as it were. And then again, as we've already mentioned, you're not wanting to use up the precious biopsy samples or things that we have, but trying to balance that, getting usable data, but with using resources wisely.
Another difficulty we have
totally
also with the donor sample. Go ahead. With the donors, the donor sample is also which tissue. For example, we need for our work, we need the vasculature, and especially the carotid artery, which they will not allow because they have to prepare the body. So that becomes a little bit difficult because the very tissues that we're trying to get is not attainable.
That depends very much on the body donor program, because for instance, our body donor program is in-house, so we can decide together with the validating doctors that receive the body first where the body will go, in which project or into teaching. So in this case, it's a sort of in-kind based principle to get the body donors for us.
Mm-hmm. So you're doing collaboration?
And we can get any part of the body or the whole body. Yes.
So you all are into collaborations for different projects?
Well, I'm a postdoc in my lab, and we would have to talk to Professor Weninger, but yes, we are definitely open to collaborations.
Thank you.
That's great. That's one of the topics, maybe in the second session is one of the goals of this meeting is to find out where they can be, what are the issues that people really want to participate in an ongoing collaborative way to try, everybody has some of the same obstacles and some of the same problems. So that's great. That's something maybe we can discuss as we go along. So what are the other important issues from each of your perspectives when it comes to obtaining tissue or tissue processing for what you're trying to do? What are the other topics that you think we should be discussing?
Mainly the IRB. That makes a big difficulty.
Yeah, that's an interesting challenge, and you know, and then you have the issue of if you're obtaining all your tissue from one center versus more than one center, it's very different now. Whether you have a single multi-center, you know, multi-center studies now are using single IRBs, and but most of you are collecting tissue at a single center, and you probably still have a single IRB. Or you have your own institutional IRB, where there can be, as we all know, enormous variability, both within the institution, but between institutions on sort of the culture of the IRBs. So what are the challenges you're having, and what would you see as potential system solutions that could go beyond your own program if we were trying to come up with a sort of system-wide solution to challenges in obtaining tissue? What are the challenges you're facing?
One of the challenges that I personally face is that the intermediaries. So you're trying to explain something to people who have limited or no scientific background. And then so it gets lost in translation while you're preparing the IRB procedure. So that is one of the challenges that happens, because there's no one who can explain it to both parties, right, before it goes to the committee. So, and then what happens when it reaches the committee, you don't get a chance to say, you don't have a say there. So there has to be some way that this transition becomes better between the PI and the people who actually will say yes or no to the project.
And are you finding you have to go back repeatedly to the IRB?
Yes, and then something gets lost in translation.
Yeah. Are others experiencing the same problems?
Absolutely. Our whole system.
Yeah.
I'll say that we actually have had a pretty good relationship and reaction to our IRB from this. And I don't know if you have this. We have, I guess I assumed that every IRB had this, but maybe that's not the case. We have sort of protocol analysts to do a pre-review, and then there can be a lot of back and forth and honing and trying to answer questions, or maybe find areas where those people have seen issues with other protocols in the past.
Yeah, we don't
to deal with that before it goes to the committee. And then the one time it sort of came back, the analyst was like, oh my gosh, I'm so sorry, I didn't see this issue. We could have dealt with this before it went to the committee. Maybe that is something that you could bring up to be implemented.
Yes.
It just depends on how big your IRB is and how much ours. We have a lot of clinical trials and different things. So the IRB committee prefers that you go through a lot of rounds of back and forth with these pre-committee reviews, because just for this reason is that it's hard if you don't get a chance to go talk with them directly. It's hard to always get on paper exactly what you mean. And then there's many things that are just protocolized or standard operating procedure that they want you to meet that you can't always keep up with because it changes all the time. And that's what these pre-review people are for. Yeah. That's been very helpful for us.
One of the things, the difficulty in that is the communication, is because now they're churning everything over online, and not like what we are talking right now. This is great, but you're just sending emails and you're sending that, and so that makes it very difficult.
I agree. I have the poor people, if they ever have the misfortune of leaving their phone number in an email to me or some sort of message, I have a list of important phone numbers that nobody wants to give out so that I can call them directly and say, I don't understand your question. Do you have 5 minutes? Because you're right, going back and forth in emails, sometimes you don't even know hardly what the question is and how to answer it.
And how many PDFs you added.
Yeah.
Other concerns that people have?
So my challenge in this area is that I guess IRB is done by my collaborator in a different country. So for us, it's going to be, we're treating this de-identified non-human subject sample. So we're going to have to coordinate with our institute, and I'm hoping that goes really well. But yeah, there's a lot of communication with our collaborators, having plans in mind for what purposes other than the initial ones those collected samples could be used, and then that would be the challenge to thinking ahead and then figure things out. in advance.
Yeah, and that whole the issue of identification is getting more and more complicated. Because clearly with, let's say, raw data from single-cell RNA-seq, you can identify someone now. And so, what is truly de-identified? And, if somebody has a relative that's in 23andMe and you have their sequence, you can often identify them now. So it really does get tricky with that. That's something we're facing that challenge right now. When we want to share the results of our data or share data online for other investigators, what do you do with raw sequence data? Can you share raw sequence data with others or not? If it was, it depends so much on the initial consents. And I'm sure it's an even bigger challenge when you have data coming from other countries or samples coming from other countries.
So when you say the raw sequence data, do you mean that individual level data as opposed to,
right.
I see.
Yeah.
Right.
Yeah.
Right, because one of our challenges, I was just going to say, one of our challenges is the goal of our consortium, which is NIH funded, is to make the data a publicly available resource for the entire research community. And there are all different kinds of users. There are users who just want the process data and have a simple question. And then there are users who are what we call power users, who really want all the raw data and they want to build their own bioinformatic pipelines and computational tools to analyze the data. And how do you protect the participant from identification, and how do you share those data, and how do you create data use agreements for that? That's something, it might be beyond the scope of this particular group right now, our charge, but it is a big question.
It'll be great to share an example of how that was dealt with in ILB. Yeah, that'll be really helpful for others.
Yeah. Yeah, so that's something that could be shared across different consortia, is how consortia, because if you get that wrong at the beginning, it really limits what you can do after that. Once you've, once your assays are done and you want to do something with the data, if you haven't done that in the best possible way, it has long-term implications for what you can do with your data.
Yeah,
So
I just want to share.
Go ahead.
We have exactly the same concern, and it has to go back. I think the consent form is very important.
Yep.
That we, depending on the languages you put it there, there's maybe certain room could allow you to share the data, but our team has discussed these issues because we are also asked to share all the sequencing data to the community to make it publicly available. And that's definitely a big concern. People, maybe not now, maybe, but in the future, will be able to assemble the genetic data and identify the individual. So it's definitely a concern.
Mm-hmm. Our IRB actually required that we add extra riders in depending on the different assays we were saying we were doing. So if, so far, since the major focus was to do single-cell RNA-seq and those things, that was under the main part of the consent form, but then we had to have a separate one for if we were just collecting whole blood to do DNA at a later time point to potentially connect to, we had to do a separate one for that. If you wanted to do organoids or any sort of propagation of cells or tissues, that was a separate consent. So in the end, sometimes these patients are signing multiple consent forms, and you're right, you have to think about that ahead of time, what you might do in the future, because otherwise then you're trying to, and we even put in there, could we recontact you to potentially consent you again for something else, because knowing that you can't plan ahead for everything, and things change so much that what you put in the consent right now may not be applicable in the future. So it is quite involved. And sadly, a very long consent form currently.
I'm trying to put together my own list, kind of organize it as from the discussion. So what I've heard so far is concerns about IRB and consent issues, and Those are largely around what can be done with the data down the road. Concerns about the freshness of the tissue for different assays and how with limited scarce tissue that's with diverse research projects in mind and different kinds of tissue, how do you make sure that the tissue is going to be as useful as it can be for the assays that people want to use? Heard a little bit of concern about what's appropriate control tissue for comparator purposes. Then if you are trying to obtain tissue from more than one center, including in some cases centers that are outside the United States, how do you standardize the tissue collection and QC the tissue for your assays? And then another topic was how do you optimize your assays prior to getting very precious tissue where you may have very small quantities of tissue and where that tissue may be more heterogeneous than the tissues that you're optimizing your assays on. What do you do about that? Particularly for disease samples, because it's, many or most of you are all working on disease processes as opposed to many of the atlas efforts that are working on trying to make an atlas of the human and health. And there it's different because, as we all know, every biosample from everybody with a disease is going to vary from every other biosample. Those are sort of what I've captured so far as the big issues that we're discussing. Other issues we should put on the table are topics that, where people think it would be helpful to collaborate or get information from other similar studies, including Human Cell Atlas, clearly.
One more point I would like to add is that once you get your data, you analyze it, you may want to look at the electronic health data of the patient. Yeah. How do we optimize that? Like, we get to that.
Yeah, and no doubt that's a big challenge. I think some of the other breakout groups are probably addressing some of those challenges of, and there you have almost the same level of issues, making sure the consents are appropriate for the kinds of data that you want to collect. Is your data identifiable, or is it going to be de-identified? What's the granularity of the data that you want to collect? And again, what's the purpose you're collecting it for? And, on the breakout group I was on earlier this morning, there was a lot of discussion about just because everybody's using Epic as an electronic health record, every center's Epic is a different electronic health record system. Everybody has customized for different purposes their electronic health record. And how do you harmonize data if you're going above and beyond your own institution? Those are all really tough questions to address. And then definitions are a big question. Something we've wrestled with is what is, how do you define cancer as a comorbidity? How do you define diabetes? Is your definition the same as another consortium? If you want your data to be comparable to some or combined with someone else's data, is your definition the same? How do you address all these questions? I see nodding heads.
It's a slippery slope that continues to get steeper and steeper. As you ask one more question, you find out that everybody is answering it differently. Yeah. And it has become very important, as you mentioned, sort of videotaping and QCing the processing of the tissue samples. We do the same thing with our nurse coordinators to try to make sure, let's all come back, are we all answering these questions in the same way? I mean, it's sort of easy to say, have they had a gallbladder out or have they had a surgical resection for Crohn's? That's pretty easy, but thinking about the medications and did they develop antibodies to biologics or did they do this or that, or what exposures have they had? It's a different, because it's not something you can just pull out of Epic. No matter what sort of schema you put, it requires actual chart review, and it can be a problem.
Yep.
for us, that's very patchy data, so we won't get that from every body donor, just some information from some of the body donors.
So going back to where we were, are there other topics we should put on the table before we start working specifically on the Google Docs? The topics I had were IRB and consent issues, freshness of tissue, control tissue, standardization across centers of tissues coming from more than one center, how to optimize assays prior to using precious samples, whether with animals or other potentially human tissue, maybe how to balance costs and quality concerns. I think, Laura, you raised this in terms of costs can escalate quickly for certain types of tissue for the assays that you want to do. Those were sort of what I captured. Are there other topics that people think would be important for us to capture coming out of this session?
I guess one that we're always constantly thinking of is, or trying to think about, is we're collecting specific samples we know for assays that we have said we're going to do as part of a project, or to try to answer the question. What are other people doing as far as saving potentially extra samples or aliquots for future, if you know, as things change? One of the things we're doing is trying to get an extra piece for, right now we're just freezing it because we don't really know what else to do. While we're in there, if we can get another set of biopsies, we do that, knowing that then you could potentially do nuclear sequencing on that, but it's not in an assay buffer or encapsulated to do single cell. But are other people doing separate things where you're keeping a piece that you don't necessarily have a plan for, but to just still have a piece that you can go back to data that samples and metadata that are well phenotyped, that you now have a piece of tissue that could be used in the future? Are people doing particular methods of saving that, for future, we're not having to reconsent the same patient and try to have another procedure? Or I don't know if that makes sense.
Yes, yes. So what we are doing is, when we get the sample from the plaque, for example, from the atherosclerotic plaque, we divide it into different sections, and one of it we always save, just in case we need to get back, and if we have to look for some cytokines, whatever. So we do that, and then because it's sectioned, and then it goes one for RNA-seq, the other for protein. So we always try to do that, and as much as possible, we try to get the FFP sample as well. So, so if you have to get back to something, then we have it.
So we're trying to get definitely a fresh, or a piece that's going for RNA-seq.
Yeah.
We're getting a piece in formalin for FFPE and those things. And then right now, for what we're able to get a third set kind of that we're just freezing because we don't really know what we might do with it. But I just want to get a sense of if other people are doing a different type of preservation, thinking ahead to other processes. Will that mean we can only do something like, regular RNA analysis, or, Western blot or something? That was really, it was just kind of, if other people have ideas of where they're saving an extra piece of tissue to maybe make it more usable going forward than just a frozen piece, which is still good. We're happy to use it, but.
Yeah, what else can we do? We have the frozen pieces, and we have whatever tissues we use for FFPs. That's basically what we can do.
Yeah.
And there's this methods development community for the Human Cell Atlas as well, where people seem to be trying to get frozen tissues to be processable for single cell data, but I'm not sure how far the people working on that are at the moment. I hope you're aware of the protocols.io methods development groups that there are out there.
Yes, definitely. So it's been very helpful that, at least in our group, the lab that's doing our single cell processing is run by Ken Lau, who is also part of the Human Cell Atlas and different things. So he's been very helpful in making sure we're trying to keep up and make sure we can use things. I was just trying to get a sense from other people if there's a different method or a different way you're preserving extra pieces of tissue, just to get other ideas.
So when you talk about frozen tissue, do you mean just put the tissue, snap freeze the tissue in liquid nitrogen and put it? What we also we haven't have enough tissue to do, but in our list we plan to fix the tissue in OCT.
Okay.
And so could be future use for immunohistochemistry, or look at if we go to the stage to look at more spatial exploration imaging, those tissue could be used for that purpose.
Okay. It's a good idea. Thank you.
We do that too with a cryo block and formalin fixed paraffin block and frozen tissue.
Yeah, and we do the same. We try to FFPE plus OCT plus liquid nitrogen. But again, small pieces of tissue, you start dividing them and you start losing some value there too. So sometimes you have to make choices. Any other topics? I think that's a good one. Future use, and what do you do when you don't know what it is you might want to do in the future with valuable specimens? Well, maybe we can go to the Google Doc then. And I tried during the break typing a few things into, I think we have a pretty rich group of prompts, if you will, and then some text around most of the challenges or most of the topics that we're discussing. Are there particular topics if we have to, well, maybe let's take a minute, and if people want to look through this, and I think if you just click on it, then you can type in if others want to add to this or organize it a little bit more. We can take a few minutes and do that. And then after that, we can pick a couple of topics or one at a time and prioritize what to put into the final templates, the blue boxes going forward. So this. I think we have good material to work with. Thanks everyone who's adding comments or clarifying text. So does someone want to suggest if we had to pick one or two priority topics from what we've been discussing, what do you think are most important that we could contribute to sort of the greater good for all the different breakout sessions?
Tissue freshness, I would think would matter a lot. So see how you can do single center studies or more elaborate studies. If we could elaborate on that, I'm interested.
Yeah, I think that's quite useful as a topic. So should we focus on that first? We can move this tissue freshness piece to that blue box. We can make that, so if we go to the blue box, the first one we can say something like for title, the importance of tissue freshness for optimizing tissue interrogation, something like that.
Mm-hmm.
As a title. So I'll just paste in, at least for now, in the blue box, this problem, tissue freshness, single center versus ship tissue. When and how is autopsy tissue useful, either as a control or for studying disease? So single center versus ship can include the sort of local versus, that's sort of a bit redundant. With local versus at a distance. Sample collection, time spent until preservation, and how that impacts, and how this impacts assay quality, let's say. And please feel free, anyone who wants to jump in and join. Does that capture the problem?
Yes, looks like.
Maybe that also, just that, definitely we want to have how this is important for assay quality at that center or within a project, but also how it impacts the ability to combine data to improve the power working across consortia. Are they really comparable? If we don't know, at least know that data, like, is it really is it really variability within the disease process, or is it just variability that we see because of how the tissue was handled?
Right. So how does this affect even tissue or biosample tissue sharing?
Yes.
And then how does this affect how to separate out concerns with or variability, tissue variability, heterogeneity,
Mm-hmm.
tissue quality heterogeneity from assay heterogeneity and quality. Is that about right?
I think so.
If that's the problem, what would we want to do to address this? And also, what would we want the larger group to do to address this challenge?
First of all, we need to set up some guideline for a standardization so that we can all work within that. And so similar kind of tissues could have certain guidelines, and that would be a better way of handling this kind of enormous workload.
Mhm.
Yes, and it could be different for different tissue types because the degradation is also different.
Mhm.
So it's tissue specific. It could be disease specific, and could be type of biospecimen specific.
Correct. And it may be going back to, say, some of the other, like the morning session about clinical metadata, how that kind of plays into it, where you can make a notation of if this sample was outside of those guidelines, in the sense of this one had a longer ischemia time or had a longer processing time. That way, at least to make a notation and that to be available, that if it's an outlier, is it an outlier because of a disease? You know, it will be hard to answer that, but it can go into the analysis of it and how it is comparable to other samples.
Emphasize importance of clinical and biospecimen metadata.
So maybe that's related to documentation. So you document how the tissue was collected or tissue was handled. Because yeah, especially clinical sample, a lot of time it's out of our control.
Right.
Okay, so we just have to take whatever they have from the operation room. So, but keep good documentation, I think, is important.
Yes, and documentation should also include where, as precisely as possible, where in the organ the tissue was collected.
And the buffer systems it was collected in, that also matters.
Yes.
Under what circumstances tissue is collected. Collected and processed. So the recommendation might be to have working groups.
Position information, if that's possible.
For what? I missed that. What was the last
Sorry,
thing?
and
Positional information, I think. Is
Oh yeah, when tissue,
yes.
Where, when. Including sort of tissue coordinates or.
Yes.
Or like
Tissue.
So in terms of recommendation here, I wonder, I don't know if it's already have some type of either like a table or listing all these important parameters in terms of tissue collection. I don't know, because at HCA already have such documents already in place, or would be something we could recommend the community to generate such guideline, but it's a list, very for like user friendly, that you can document that all the tissue you collected. So once we have that standard form, kind of that, then people are easy to know, because otherwise everyone has their way of document the tissue. So if the from NIH, from Human Cell Atlas could develop such kind of a standardized form, then everybody is easy to follow, and that will in the future will allow better comparison of the samples and data.
Or an app can be developed so people can access it and input their data. And so we can, like sort of have an understanding what's going on.
Does this capture that? Establish standardized tables across various consortia to prioritize metadata elements, make an app that others could use?
Mm-hmm. Mm-hmm. Yeah.
Just speaking of that, just curious if there's already anything in place about like kind of hotline chatting channel or something, because I guess it could be just me, but people don't know what's already in place and what's needed to be done, because it's also kind of a method of communication with people who are already deep into the process, and we can easily get some information for some certain things.
So hotline chat channel, or maybe establish a hotline chat channel for different researchers to communicate about common everyday problems. Does that capture that?
It could be included in the app, makes it easier. Otherwise, people have to go different places. So it should be consolidated kind of thing.
Okay, so this can be connected with the app.
An easiest thing would be a dedicated Slack channel or something like that. That doesn't need a lot of work towards that.
Yeah. For example, a dedicated Slack channel. I think we're doing pretty well.
Mm-hmm.
Any other recommendations for this problem? Sort of initial tissue freshness and processing question, really.
We could also have training programs based upon this, so that if you have postdocs, others who will continue work on this kind of project, they already have a sense.
We could say cross-cutting training programs for postdocs and others. And others working in this area, something like that.
Mm-hmm.
I think this is pretty good. And everyone can put their you can list names. Gian, you're our note taker. We'll put you down here. Our volunteer. Yep. And everybody else can add their names.
Do we include our emails here or not?
I think the organizers have our emails, but it's certainly okay to include them. We have about a half hour left. We could, if we choose, pick one more topic to focus on and as a priority. One that I thought I heard that might be good is the combination of the issues around the IRB, around consent, around identification, and upstream, making sure upstream at the start of a project that you're not decreasing possibilities for data sharing or maximizing how data can be used, clinical data or other types of data, nucleic acid data that might be make someone identifiable, kind of combining all those together. I see nodding heads. It sounds like we may have consensus to take this one on.
Yes.
So title could be Challenges with Informed Consent. Patient identifiable data. If I can spell. Oops. And potentially patient potentially identifiable data. We can probably cut and paste from above. Let's see. So any thoughts on what recommendations would be? The idea, of course, is we're not solving the problems now, but we're identifying areas for collaboration to either solve problems or so groups can work together.
So as Laurie mentioned earlier, how they do the communication, that would be a helpful start. With the
Hey,
IRB
Laura,
teams.
do you want to rephrase? That was about, was that about communication with your IRB?
Yeah.
I mean, I do think that one of the things that maybe could go forward be help in, if there's a way to draft some at least boilerplate guidelines for data sharing across consortia. Potentially if someone who's already been through at least their local IRB process, clearly that might still have to be edited to meet each individual IRB. But at least we'll give people who haven't had to go through that process specifically before a template for where to start from, the bases you need to cover. And what's required in that, depending on the type of data that you're trying to share.
Data share across centers applicable to most consortia or most research studies and consortia.
Mm-hmm.
That would be acceptable by most IRBs or by that would be considered acceptable by IRBs in general, something like that.
Mm-hmm.
So that everybody doesn't have to reinvent the wheel with their application.
Right.
And if someone has a specific question, they could be helped by this group who is coordinating this maybe.
So then you get back to this issue of a chat room or some forum room or forum for people to, diverse investigators to discuss challenges they're having, something like that.
Mm-hmm.
To address challenges and describe best practices they are facing and to describe best practices, something like that. What's working? In some ways, it's similar here to that. Well, it's the hotline channel idea.
And some way of really understanding consent, because you have different write-ups for different protocols and different hospitals. How do you really have a clarity on that?
Clarity on what is meant by consent.
Mm-hmm.
What are the implications of different language? Consent process for downstream use of data, something like that. Does that sound good?
Sounds great.
Any other recommendations?
I wonder for such guideline or even template, are we able to obtain this from, say, the NIH or the Human Cell Atlas? Could be more, because now everyone's doing it by its own institution, right? But we don't have a, I know here it's just a, do we comment for certain guideline, but I don't know if like NIH, is it possible for NIH to develop some type of more official language? I may be thinking too big, but just for everyone, because I feel that this type of discussion I have in my own small group and it's here,
Right.
and so everybody struggle, right? Yeah.
Right. So you may be thinking too logically and rationally. You know, it is a challenge because of, you know, how federated the different institutes are at the NIH. And, you know, it would be great if we could, you know, if the NIH would take this on and create a template for all studies, and then the IRBs from, you know, everybody who's part of the CTSA program, which are, you know, many of the IRBs. And now with the central IRBs that are being required for most studies, if they could all adopt the same language or have a menu of different choices that were acceptable, and people knew up front that they would be acceptable for different purposes and different context of use, that'd be great. Maybe we can recommend that.
Right. Right. We just recommend and see.
Right. No harm in
Maybe
recommending.
that's, yeah.
Is it
Right.
possible?
Right.
Yeah,
For the NIH?
some of that might be local legislation based too. So
Yeah,
I
there
think that
are
we cannot standardize everything, but it would be good to have a guideline to start with.
Right,
Yep.
right. There are state regulations that do
Yeah.
affect this. But is it possible for the NIH to take the lead across all institutions, all institutes?
Mm-hmm.
And I think the NIH cannot solve globally the problems about communications and all those kind of logistic problems, but at least there should be some centralized efforts to kind of define what's de-identifiable, what's identifiable, because with the advent of new technologies, the definition keeps changing. So at least at the forefront, somebody should like lead with some examples, and we'll benefit from that.
Is it possible for the NIH to take the lead across all institutes in designing a centralized effort to define what is identifiable data? Can the NIH work with the CTSA program and single IRBs to come up with a universally acceptable templated language for data use. Something like that.
Mhm.
Yeah, great,
Yeah.
thank you. Yeah.